We're starting the recording. It is July 28th. And let's start the week how we always start the week, where we look at the schedule. I'll also let you all know I do have your lab and lecture midterms. We'll end lecture a little bit early today. I'll try to end 10 minutes early, so you have time to come up, look at them, see what you missed, see what you got right. And sometimes, you can even argue for points. If you're like, I don't think you understood what I was trying to say in my answer, I can be like, oh yeah. Or if you say, the midterm, I found it misleading in the lecture question. Sometimes people do that, and everybody gets points if you can demonstrate to me that the question was, unfair. So take the time, come on up, look at your stuff, and don't be shy. I won't take it personally if you say that a question is unfair. That's my promise to you. All right, here we are. In week 4, what do we have? We've got essentially cell cycle stuff. If there's one thing that the upper division biology courses want you to know when you leave this class, it is cell cycle. Every faculty meeting where they talk about the curriculum, they're like, the students don't know the cell cycle when they get to my upper division course. And so it's like, fine, we'll do more cell cycle. So this entire week is mitosis and meiosis effectively. All right? So you have one assigned chapter reading, and then we're going to get into the stuff of life as your assigned reading. As I've mentioned, there's a scanned PDF of the book. I've also got a few copies that I could loan out if you want to borrow a hard copy. Yeah, it's really great. And if you don't want to do that, the reading recordings are posted in the module for the associated chapters from the textbook. Make sense? Question?
Do you know approximately what page number, page range for
The stuff of life?
Yeah.
That's really like a 1 to 2 sit down read. So you want to just read through probably the whole thing this week, and then read through it, then review it a little bit next week. All of my questions are still based on lecture slides and textbook readings. You'll just find that if you do that reading of the stuff of life, that you'll understand the stuff. Like, oh, I get it. I didn't need to read the textbook. That's at least my hope. Yeah, but how about read half this week, read half next week? That'd work too. Cool. Good deal. This week we have 3 labs. This is our last full week with no midterms or anything. So we've got DNA extraction today, bacterial transformation tomorrow, and then we'll do this is a dry lab, the population genetics and Hardy-Weinberg one, where I give you an additional lecture about Hardy-Weinberg equilibrium and population genetics. It's kind of dry, and then there's an online, essentially a full online lab that you do as your assignment. Because this is another concept, Hardy-Weinberg equilibrium, that gets lightly touched on in probably 3 or 4 different courses that biology majors need to take, and you never get, in my opinion, a full explanation. So I'm gonna hit it, I'm gonna double tap it, right? Pop, pop, and we're gonna really make sure that Hardy-Weinberg's stuck in your brains. Any questions about this week? Okay, stupendous. I put a little note in the course module that if we wrap up our week 4 PowerPoint, we will just start jumping into the week 5 material. Week 4 PowerPoint's only 50 slides long, but sometimes in the summer, that takes us the full 4 days. Sometimes in the summer, that takes us 2 days. So we get more flexibility in the summer, Okay, I'm just double checking. We are recording. My microphone is working. And we're ready. Yeah? All right. Summer session, week 4. Here we go. The cell cycle. So up until now, we've been, we started with chemistry review. We zoomed out to look at the cell overall. We focused on the membranes a lot because those are the fundamental units of organization, right? They separate the different chambers. We talked about energetics. We talked about enzymes because the enzymes are essential for determining what's going on in those different chambers that were set up by the existence of the phospholipid bilayers. We focused on cellular respiration and photosynthesis because they are the foundation for the whole energetic cycle of the planet. That required a discussion of some organelles. Now we're talking about making new cells. Okay, so cell division. Why does it need to happen? Well, unicellular organisms reproduce by cell division in a process called binary fission, where they go from one thing and then they split and they make two things. All right? This is clonal reproduction, and if you're reading the stuff of life, it's the foundation for the storyline in that graphic novel, right? That there's an alien race and they only reproduce asexually. But this has caused a lot of problems for them because now they're dying of these diseases, and because they're all genetically identical, they don't have any unique resistances to the diseases. So one thing gets sick, everything gets sick, right? So asexual reproduction, it is an option, and the problem with it is that it doesn't generate variability. Now, I'm going to take the time right now to talk about this bullet point right here. If a single-celled organism is going to produce two identical copies of itself, it needs to do a number of things, right? First, it starts out small. It needs to get a little bigger so that when it splits, each, what we call daughter cell, has enough material to survive on its own. All right? When we're talking cell division, we use the language of the mother cell and then the daughter cells. Okay? Also, all the genetic material needs to get replicated. Okay? So if we're focusing on unicellular organisms, prokaryotic mainly, I guess, in this case, you need to reproduce all that genetic material, okay, so that each daughter cell has a full set of genes. So far, so good? Now, as you know, there are eukaryotic unicellular organisms as well. And so those eukaryotic unicellular organisms are also going to need to make some extra mitochondria or chloroplasts so that when it divides, each daughter cell has mitochondria and chloroplasts. So this is the big key point, and that's why I'm hovering and waiting on the first bullet point of this week, okay? Which is the daughter cells need to have enough to live on their own. And the key thing, the key point here, is that they need to have all of the genetic material. Because we're going to get into a whole discussion about DNA replication, and it's complicated and it's essential. Otherwise, you make a daughter cell that doesn't have the right genes, doesn't have all the genes that it needs to survive. Okay, so in unicellular organisms, we're talking just reproduction. In multicellular organisms, development, growth, repair, renewal, okay? Sperm, egg, they hit single-celled zygote, right? Yeah. It divides a bunch to develop. As the organism ages, it develops new tissues maybe, okay? It changes, it gets bigger, it develops, it grows. In the organism's life, it takes damage, right? And it must repair itself. And you repair via cell division as well, okay? So you've got, you burn your, I cut my thumb this week, this weekend. And you burn yourself on the grill. I had a barbecue, and so I'm thinking about grill stuff, all right? And in order to repair, cells need to divide to replenish those now missing cells. We'll talk in very general terms about how the cells do this, okay? It's all about just sensing if there are cells next to you. And if there's no cell next to you and there's supposed to be, just divide, make another cell, fill in the gap, right? So development, growth, repair, and then renewal is very, close to repair, but renewal doesn't require damage, right? Your cells are constantly renewing. You've probably, okay, there's no way this is stood the test of time. But back when I was an undergrad, all the heady kids would watch Waking Life. Yeah, okay, cool. We got one. Yeah, nice. And in that movie, they talk about the whole, oh, you're a totally different set of cells than you used to be. You know, so every 7 years, you're fully a new organism. So how can you still be you, man? Right? So your cells renew naturally because cells get gunked up and crummy as they get older. And this is essentially what aging is when you lose the capacity for cell renewal and you have to keep going with older cells. All right? And this is why most attempts at reversing aging or increasing longevity have to do with reinitiating cell renewal. Okay? We'll talk about what limits cell renewal a little bit, but it's very interesting to think about. Okay? Cool. You know how I feel about these videos. This one, I'm actually not gonna play because on a Monday morning, I don't feel up to it, okay? But it's embedded in your PowerPoint. And trust me, I'm gonna go over this stuff a lot, all right? And maybe later we can play it if you all really wanna see the outdated CG. Okay, mitosis. Every semester, I have to say that I keep mitosis in my soxus, and it's very funny, and we all laugh, and we have a great time. Okay. So mitosis is replicative division, okay? It takes a cell, and it makes 2 identical copies, all right? That's what mitosis does, all right? Mitosis are also essential for balancing. Very good. You make 2 identical cells. They could be haploid, they could be diploid. Now, ploidy. If you've taken Botany 105 before this course, you've discussed ploidy quite a bit because you have to go through all of the life cycles of the different types of plants and whether the diploid or the haploid phase is dominant and what polyploidy is and all this stuff. But if you haven't, then I don't think it's fair to not introduce haploid and diploid right now, all right? So ploidy, that's the general term for it. Ploidy, okay? And there's gonna be a slide that goes over this again later, but you know how I am. I repeat stuff a lot that I think is important. It's the number of copies of the genome, okay? The genome being all chromosomes. This is pretty accepted shorthand for chromosomes. If you're in bio, you have to write the word chromosomes a lot, so people will do this ohms thing, but that means chromosomes. If you remember, bless you, procs have 1, eucs have many. All right, 23andMe, the service that sequences portions of your genome looking for important genetic markers and identifiers, all right, is named after the number of chromosomes you have. But you are a diploid organism. Therefore, you technically have 46 total chromosomes, but being diploid, you have 2 copies of each, all right? So we talk about ploidy like this. N, haploid, 1 copy. 2N, diploid, 2 copies. And these can go up. There are triploid, tetraploid, octoploid organisms, okay? But in Bio 105, to try to keep it as simple as possible, we focus on the human cell cycle, which just involves diploid, okay? I'm gonna make a little note down here. Polyploidy exists, It's way more common in plants than in animals. All right, plants seem to be able to handle having more copies of their chromosomes better than animals do. Okay, so now that we've talked about ploidy, are there any questions about ploidy? Think about it this way, right? The sperm cell had a full complement of chromosomes, 23 chromosomes. The egg cell had a full complement of chromosomes. I'll do them to scale, all right? The sperm cell had 23 chromosomes. The egg cell had 23 chromosomes. They came together and they made an organism with 2 copies of each chromosome, a diploid organism. Cool? All right, but we'll still say that your chromosome number is 23 because there are 23 unique chromosomes. How's that feel? Is that okay for everybody? Cool, 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 cool. All right. Mitosis makes identical cells. It's called replicative division. Okay? Meiosis reduces ploidy. It's called reductive division. All right? Mitosis is what we're going to start with. Take one cell, make 2 identical daughter cells. They won't be identical in, say, the size, because they were bigger and they got smaller. They won't be identical in the number of mitochondria or the number of chloroplasts. But the genetic material should be identical if nothing went wrong. That's the goal of mitosis, to make them identical. So when I say, oh, the cells could be haploid or diploid, what I mean is if the cell was haploid to begin with, it makes haploid cells. If it was diploid to begin with, it makes diploid cells. If it was tetraploid, it makes tetraploid cells. They're copies. How we doing? Good so far? All right. Now we've got meiosis tied to the life cycle because meiosis is what makes gametes. So sperm and egg cells, or if you're not looking at humans, microgametes, megagametes. Some things have gametes that are not different in size. They're pretty identical. They have isogametes, right? But that's not something we're going to focus on. We're going to focus a lot on us, humans, because we're the best. We win, we won, we dominate, right? Never done a bad thing. Somatic versus germ cells. Let's talk about them. Okay, another quick check-in about what students think is cool and groovy nowadays. All right. You all reading Brave New World? You got to read Brave New World, right? Solid. You have to read 1984 in high school, and then in college you read Brave New World, and you go, oh, Brave New World is like 1984 for college people who get it, man. And then you read other stuff, or you don't, because, I don't know. It's hard to read. There's too many letters. It's frustrating. The drug that they take in 1984, in Brave New World, the drug that is given to the citizens to keep them docile and just content is called Soma. Aldous Huxley's least father was a pretty prominent scientist, so he uses science words in his writing. All right? Soma, somatic cell. You think about giving soma to the population to make them all just part of the larger body and just make them docile and play their role in society. Well, that's kind of what somatic cells do. The majority of your organism is somatic cells. They're diploid cells. They make up all the organs, all the tissues. It's like everything. Over 99% of the cells in your body, okay, are somatic cells. They carry out mitosis. They make copies of themselves. They regenerate. They replenish, all right? They're somatic cells. Now, this can be complicated by the fact that there are pools of stem cells in your body that replicate to make undifferentiated cells that then can sort of differentiate to make different things, like the cells in your bone marrow. They can become different cell types. There's still somatic cells, though, which will make sense when we look at the definition for the germ cells. The germ cells are specifically involved in producing gametes. So they are specifically involved in dividing by meiosis and making sperm and egg cells. All right? Good so far? The cells that undergo mitosis are the somatic cells, is another way to think of it. And the cells that undergo meiosis are the germ cells. All right, you've got a very small pool of germ cells in your body that divide via meiosis to make gametes. The reproductive cells. They will have half as many chromosomes as somatic cells, but they will still have the full complement of chromosomes, right? They've just gone down in one copy of the genome. They'll still have, in our case, 23 chromosomes, but there won't be 2 copies of each chromosome after meiosis. How are we doing so far? We're just laying groundwork, and then we're going to look at a bunch of diagrams about how these processes happen. We go through mitosis because it's simpler. Then we go through meiosis, and then you'll understand the cell cycle. Cool? Cool? All right. There it is. Next slide. Here we are. When I was young, we still used overhead projectors, and the teacher sometimes would have to have somebody at the back running the slide machine. You know, the projector is what it's called. And they'd have to go, next slide, and the slide would come up. It's a simpler time, by which I mean more complicated and worse. It's a worse time. The genome. All the DNA in a cell constitutes the cell's genome. Okay, fine, you could say, well, wait, what about my gametes? Does my gametes have a genome and the organism has, and the other cells have a genome? And it's like, yes, all right? Usually when we're talking about an organism's genome, we're just talking about all the unique chromosomes that it has, all right? So this right here is called a karyotype, this image. And this karyotype is of a human genome, all right? This individual has 2 X chromosomes. You can see them right here. 23 different chromosome pairs. This is from a diploid cell. The chromosomes do not glow like that naturally. These have been colorized. I believe they do these with, they stain all the chromosomes in a bunch of different cells. They splat the cell, they lay them out, they find the right one, they take a picture, and they constitute an image like this. They don't take little microscopes and probes and lay out all the chromosomes, all right? A lot of Photoshop to get all of them to line up like that. Okay, so this right here is a human karyotype, but not every karyotype with 23 chromosomes is necessarily a human karyotype. I want to write that one down. It might be on an exam. Okay. Not all organisms with 23 unique chromosomes are human. Organisms vary in their number of chromosomes drastically, even between closely related organisms. Okay? Cool. Moving forward. In the prokaryotes, the entire genome is a single circular chromosome. All right? It's just a big loop. The prokaryotic genome is very tightly packed with genes, all right? There's not as much regulatory junk going on there, okay? I'll explain more of that as we move forward with our genetics discussion. Over here, diploid organism. You see 2 copies of every chromosome? Cool. And look, the chromosomes are numbered based on their size. Cool? Please. Yes. Chromosome numbers are determined by size. It's a solid question. I'll give you 1 John point for that one. Yeah. Yep, that's the convention. Yes. Please, please. Absolutely. So here's the idea, right? We've seen N before, right? When we looked at, let's think way back when we talked about carbohydrates. Remember C6H12O6? Carbohydrates have C sub N, in parentheses H2O sub N. That's like their formula, right? This is meant to indicate that this number could be 23 chromosomes, it could be 12 chromosomes, but in the haploid cell and the diploid cell, they both have the same unique number of chromosomes. That make sense? Awesome. Great clarification. 1 point for you as well. All right. Cool. Oh yeah, and the chromosomes are linear in the eukaryotes. It's like a massive headphone cable that you bundle around a little headphone cable carrier, which is an out-of-date thing, but people still use them, right? You gotta have your backup wired headphones. So check this out. Man. I'm kind of, whatever. I was kind of proud of myself I was thinking about, I gotta discuss chromosome structure. And then I put the slide about chromosome structure from the Stuff of Life on here. It's okay to be happy with the work you do sometimes. But you don't have to tell everybody. I'll keep it to myself in the future. Anyway, this is just, for example, what you'll see in the Stuff of Life book. And I can just easily zoom in. So when you saw earlier that the chromosomes in a eukaryote are linear molecules, okay? Well, here's an illustration of what that sort of looks like. They're very long, stringy, linear DNA molecules, okay? But in preparation for cell division, they clump up, all right? They clump up like what we saw on the karyotype. So you're going to see in all these diagrams the DNA, just like they say in this book. As a visual aid, they stick with the mitosis-meiosis form as they talk about chromosome anatomy, okay? But most of the time, the DNA is in a long, linear, spread out form because it's got instructions on it for proteins, right? It needs to be sort of spread out to be read. It clumps up so that you can more easily say, I'm about to divide, this chromosome goes there, this chromosome goes there, this chromosome goes there. Cool? Question.
Is that assuming that a better picture would be that, 23 chromosomes, that it's one continuous chain rather than distinct chromosomes?
They are distinct still. The example that I always use, imagine you've got, just bear with me, we'll see if this, I'm gonna use a new analogy. We're trying something new actually. We're not gonna use my classic example. Ready? You have 23, you have 46 pieces of wire. There are 2 red pieces. There are 2 green, 2 yellow, all throughout all the colors. You put them into a bucket and you shake the bucket around a bunch, okay? Now you've got a jumbled up mess of wires. And each wire has a little story written on it. Okay, so you can get in there and read the story. But when it's time to make 2 boxes, which each have just one of every color of the chromosome, it'd be nice if those were all wrapped around and bundled up. Like wrapped around a little screw or something. Then you can just take the screw and be like, oh, the red one, boop, red one, boop, green one. See what I mean? So they don't join into a long chain. They're still separate molecules the whole time. Unless something goes on where there's like crossing over, there's transpositions of pieces of chromosomes to other chromosomes sometimes, which messes stuff up. And sometimes a chromosome will split when it's in the linear form and it won't kill the organism, so then the organism persists. Like, there's so much to it. Especially when you think about the fact that we've got 23 distinct chromosomes. And the original life forms had 1 circular chromosome. How to go from that to that. A bunch of snapping, problems in reproduction, replication, make an extra one here, break pieces off, they go all over the place. It's all super complicated. Okay, so here's a chromosome in the mitosis-meiosis sort of framework right here. Okay? A few things are indicated on this chromosome. I'll show them to you again with sort of textbook images, but if you can't tell, I just love this book. I don't get money when they sell copies of this book. But boy, oh boy, I sure would love it if they ever made the ones they were planning on making ones for all the different fields, and they just didn't do it because nobody bought Stuff Alive. Boo. Okay, cool. Here's a chromosome, all right? The chromosome contains genes. The genes are instructions for making proteins. We've talked about that before. Remember the central dogma discussion, okay? The location where a gene is at is called a locus in the singular, loci in the plural. So this is the locus for gene A. This is the locus for gene B. This is the locus for gene C. There's a pinched region in the center called the centromere, which is very important because later on, microtubules are going to attach to these chromosomes and move them around for cell division. Like if I'm taking the bucket of wires, my hands have to grab onto something. Let's say that the nut is magnetic or the bolt is magnetic. So I can just boop, stick a magnet in there and pull the individual wrapped up chromosomes out. This here, this centromere, is super important as an attachment point for when the chromosomes need to be moved around during mitosis and meiosis. Because remember, the chromosome's going to bunch up like this. This is what it's going to look like when it's time to move chromosomes around. Okay? We will talk about telomeres a little bit. But the end of the chromosomes have these non-encoding regions, non-coding regions, all right? It's just to protect the DNA. And don't worry, I'll explain that in more detail. Now, before a cell divides, it has to make double the DNA, because the daughter cells each need to have the same amount of DNA as the mother cell. Are we cool with all that? So it replicates, and it looks like this. Okay? This is a replicated chromosome. This is an unreplicated chromosome. This is a replicated chromosome. They're both chromosomes, so the language can get really confusing. When you're looking at a replicated chromosome, it sort of has these two arms that should be identical to one another, as indicated here with the same letters, and the letters also being lower and upper case at the appropriate locations. You see? Okay. Cool. So don't worry, if that didn't work for you, here are your textbook images. This is I only use Stuff of Life in the summer. Fall and spring get this. So I'll give you the fall and spring stuff too. Okay? Here's an image of DNA condensing. All right? It starts out as a bunch of spread out spaghetti. It condenses into these clumped up chromosomes. Here is a replicated chromosome. It has a centromere, which will be an attachment point for microtubules to move the chromosome around. It has two sister chromatids. So once a chromosome replicates, unreplicated to replicated, each arm should be the same if the system worked correctly, right? Cool, 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 cool. So each arm is called a sister chromatid. You want to get really confusing, when the DNA is unreplicated, it's called chromatin. It's ridiculous. All right? So chromosome, unreplicated chromosome, replicated chromosome, and then they'll get yanked apart, and now it's two unreplicated chromosomes. Chromosome, chromosome, chromosomes. Okay? Cool. When there's so much overlap with terminology, I'm really going to emphasize it because you want to use the right word. Another check-in, these US students, you still watch Into the Wild, and then they like shockingly show you his penis when he's floating down the river, and it's like, oh my God, you know? Yeah. He talks about that in that movie. He's like, the beginning of understanding is knowing the right name for things. And then he eats the wrong plant and dies, right? Yeah, so it's pretty cool. So the beginning of understanding is knowing the name for things. It's really important. It's why we actually emphasize terminology in science courses. Once you have the terminology set, you can discuss things in more depth. So when you hit genetics, if you decide to keep going that route, you won't be all stuck on, oh wait, is it chromatin or chromatid or sister chromatid or replicated or when's it a chromosome, when's it not a chromosome? I don't get it. Why is there 2 phases of meiosis? You'll be able to deal with the complicated stuff because we're going to lay the foundation here. Anyway. More of the same. Just in case this is helpful to you. It's just a different sort of diagram. Check it out. This is going to happen to all of the chromosomes in preparation for mitosis. All right? All the chromosomes are going to condense. The DNA is all going to get replicated. All right? Making 2 sister chromatids on a replicated chromosome. Then they're going to split. Centromere was the attachment point. These got spread out in the formatting a little bit, which might be confusing. So my apologies, because you can see here that the DNA is duplicated. The DNA is duplicated here, but it's replicated before this process. I kind of actually, now I'm looking at this, I hate this slide. There's a difference between replication and duplication. All right? In the replication step, okay, you're making new copies, okay? In the duplication step, you're getting the sister chromatids. I'll explain it to you, I promise. Just don't get caught up on it yet. Here. Cell cycle will make you more comfortable, I promise, yes.
The chromosomes are all part of the same DNA strand, right?
Each chromosome is a distinct, separate strand. Yep, so it's like the big box of unwound wires, all right? They're unique and distinct, and you can pull them out and separate them. But when it's time to actually separate them effectively, they get bundled up, so they're easier to move around. It also means that during this part of the cell cycle, you can't really be you can't really be reading the DNA because it's all been wound up. It's like a long strip of paper with instructions on how to make lactase on it, right? Once you wrap it around your finger, yeah, it's easier to move around, but I can't read the instructions anymore for making lactase. Make sense? Sick. But to answer your actual question, they are each distinct linear molecules that then condense. Did that make sense, or did I muddy the waters further?
They're not part of one single strand, they're each their own.
Yes, each chromosome is a distinct strand. Yep. Oh, sick. Oh yeah, let's go back to the locus. This is great. All right, so it's a long strand, and when it's spread out, this could be locus A. And this over here could be locus B. All right? It's still the same chromosome, and this is actually really good to lay out here. Diagrammatically, we always, it's an issue in in biology in general. We always present DNA like this because it's easier to represent than a big long strand with a big long portion that's like color-coded red, and that indicates locus A. Instead, we look at it in its condensed form. So this is all bunched up right now and isn't really being used to make gene A, all right, or protein A encoded by gene A. That was locus A right there, where gene A was, and then it just got all condensed up. This convention here, and as you'll see again here in the textbook, is just to make it easier to present with diagrams. But during most of the cell's life, the DNA is spread out like this. How am I doing? I got you, then you, yes.
Clarify, it's like if you got a whole bunch of different scraps of yarn
Yeah.
rolled into a ball, not one ball of one piece of yarn.
It is more like one piece of yarn rolled into one ball. Like you've got a bunch of different pieces of yarn, and they're. Is it? I don't know much about yarn. Oh, wow. Okay, it's like a variegated yarn. Apparently, no, they are not. These are not attached. And when they condense into one. Oh, I get it. I know what's going on. I totally understand now. I think what's happening is there's only one chromosome here.
Yeah, and it has parts.
Yeah.
Or genes in it.
There's actually not only one chromosome here. In the cell, there should be 23 chromosomes here. And there should be 23 of these. And then they should split, and there should be 23 of these.
You got like 21 individual strands of yarn put into one ball.
They do not go into one ball. Each individual piece of yarn gets rolled into a ball.
Yeah, 23 strands going to 23 balls.
23 strands to 23 balls, and then the balls can get, and then,
The balls are variegated.
yes, but each, it's gonna be like two strands like that are woven together in the ball now. That's variegated, right? And then you're gonna yank those apart. It's very complicated. And I think that our big problem is there is not only one big chromosome here. Let me show you an image. I think it might help. And then I have not forgotten, I got you, and you both have questions. Okay, check this out. Here's an actual picture of this process, all right? So the chromosomes are all uncondensed in a big blob here. It's just a big box of yarn right here. Then the chromosomes start to condense, okay? So now each strand of yarn got sort of bunched up into a ball. Now they're fully condensed. See what I mean? And so there's 23 distinct condensed balls here. All right? Does that kind of make sense?
Oh, think about it, they're more skein shaped.
They are skein shaped. I know what a skein is. They are shaped very much like skeins. Yes. Then the sister chromatids will be pulled apart by microtubules. And then the DNA sort of starts to uncondense. You'll see this a lot of different ways because it is incredibly complicated. I got two ahead of you. Yes?
So I think you said that one picture with the locus, you were showing locus A and then gene A, so those are the same. Are those synonyms? Can you use those interchangeably?
No. The locus is the location. The gene is the sequence of DNA. So the gene exists at the locus, but the gene is actually a sequence of DNA instead of a location.
The sequence of DNA, when it comes to expressing genes, is that like that protein that expresses one thing specifically, or can it do multiple?
Yeah. There's go ahead.
One correct producer, the locus is the individual trait that is expressed, and it's on the gene.
That is oftentimes the case. Yep. So the gene will usually, and I want to stick with that right now, and then later we're going to kind of disrupt it because there's a whole thing where this gene is going to make an intermediate molecule that then gets chopped up and mixed up. So then it could technically make different things, right? But you are correct as well in that the gene should be encoding one thing, right? So the gene is the actual sequence of A, T, C, and G, right? in the whole sequence. If you read with a molecule called RNA polymerase, you will make an RNA molecule, which then can be read by a ribosome in order to make a protein. All right? The locus is just a term for the location, which can be important when you're talking about chromosome structure, and you're like, oh, wait, this is the locus that gene A usually resides at. But as we're looking at this sequence, this genotype that we've developed, the sequence is different at locus A. So it would be like the drawer here always contains the instructions always contains my answer key. It's not in here. These are different documents, okay? The drawer here always holds instructions for how to make a bicycle, all right? That's locus bicycle. This is gene bicycle. So let's say one day you go and you look in locus bicycle, but you don't find gene bicycle. So that's why it's important. Sometimes it feels like, why do we have to have different words for these things? But that's why, because the locus is the location, the gene is the actual sequence. Is that okay? Solid? Solid. I like it. I had you as a question as well.
A good follow-up to that. Is there kind of a fun, just for fun question, is there like some cool physics chemistry reason that we can say so confidently that the locus always indents, that it basically the string always wraps up in exactly the same way?
There may be, but my understanding of it, my level of understanding of it, is just that it's because of the sequence on the linear strand. So the linear strand gets bundled around these histone proteins that then sort of associate with one another and get bundled around additional proteins. And because it's like a curly fry. You know what I mean? You extend the curly fry, let the curly fry strap, or maybe a spring. I'm just hungry, I guess. You know?
So like the sequence itself determines how it curls.
Yes, and I can tell you with almost 100% certainty that a geneticist would give you a different answer to that question. But that's the level of my understanding. Yeah. Okay, yes.
I forgot.
If it comes back, don't hesitate. That's why this PowerPoint is 50 slides for 1 week. You know what I mean? Because there's a lot to discuss in it. And I'm really, really happy that people are, asking questions. Okay. So we're going to rapid fire past this. And if you are holding in your head the whole, let's think about replicated versus duplicated, please don't. All right? I shouldn't have said anything about that. Just mark it out of your notes. We'll just pretend I never said it. All right? Let's talk about the cell cycle. And if that stuff so far didn't quite make sense, there's a good chance that the cell cycle will explain it. So we're going to give it a shot. Here we go. Most of the time, the cell exists in what we call interphase. All right? Interphase is when the cell is doing its job. The DNA is uncondensed so it can be read. You're making the proteins you need. You're carrying out whatever. You're carrying out respiration, maybe photosynthesis. You're generating enzymes. You're doing all this stuff. You're making lysosomes and breaking things down. You're doing all the stuff that we've talked about, all right? Interphase. Interphase is the majority of the cell's lifespan, okay? It prepares the cell. Also, there are steps of interphase that prepare the cell to divide in what we call M phase, all right? So if we're talking about mitosis, the M stands for mitotic. But if we're talking about meiosis, the M stands for, I believe, meiotic is the word. It's always M phase, so it's okay to just learn it as M phase. Not going to be a problem for you. All right? Cool. Interphase. Cell growth and preparation for cell division. That's only if you're looking at this from a cell division-centric perspective, right? Because it's also doing all the things that you need the cell for. Cool? All right. Within interphase, there are 3 stages. The stages are G1, S, and G2. All right? Sometimes these are taught as go, stop, go. I like to teach them as growth, synthesis, growth, or go, synthesis, go. All right? S is when the DNA is actually being replicated. And the reason it's taught as go, stop, go is during DNA replication, some cell functions, normal cell functions, or some normal transcription translation functions can be slowed or stopped because the DNA is the focus is on replicating DNA at this point of interphase. Question, please.
What did you say your preferred GSV acronym is?
I like growth, synthesis, growth, because the cell grows in G1 and then it synthesizes new DNA. Oh, it synthesizes copies of the DNA and then it grows a little more. And that's it. And remember, the cell needs to be growing because it's going to split, and the 2 daughter cells need to have enough stuff to survive on their own. Okay. How do we feel so far? We're not going to talk very much about GSG, but you have to know what I've just sort of laid out for you. That the cell is growing, okay? Doing its regular thing. The cell is growing, doing its regular thing, but there's an intermediate stage called the S phase of the cell cycle, okay? Where the cell is duplicating all of the chromosomes. Okay? How we doing? It's pretty, that's the reason that I'm hesitating right here is because we're going to be focusing on M phase. And I'm just going to be assuming that, oh, you get it, the DNA is replicating in S, it grows, S is in the middle. Yes?
So the duplication happens in S phase, and that's in preparation for all the steps of mitosis. So duplication does not happen during mitosis?
Yes, 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 yes. That's the key takeaway. You get 3 John points. Very good. And everybody hear that? That's the key takeaway here. That the replication, the DNA replication, does not happen during the M phase, the mitotic phase. All right? When you enter mitosis, the DNA is already replicated, but it is not condensed yet. And because it's not condensed yet, you can keep using it during G2. All right? But as you enter into the M phase, all the chromosomes are going to condense down. All right? I got 1 ahead of you. Yes?
I was kind of curious if we ever talk about, I guess, the S phase, specifically about how, I'm curious if we'll go into DNA replication at the same level as the whole photosynthesis process in terms of where the electrons come from and where the molecularly how that process comes together.
We will talk about it molecularly, how the process comes together. We've got plenty of DNA replication stuff. It's a really important process, right? So yes, the energetics of it are for our level surprisingly simple. And I'm just so excited by your question. You're gonna get 2 points. And I'm gonna tell you that, remember ATP? And I told you how, it's just like a nucleotide, right? But it's got the extra phosphate group. And it has the energy to drive anabolic reactions, right? To synthesize long polymers. So what if the free-floating nucleotides, A, T, C, and G, all exist in solution with that extra phosphate group on them, or those extra 2 phosphate groups? Well then, wouldn't they contain the energy needed to synthesize their own polymerization? And the answer is yes, they would. So the enzyme that's going to be responsible for snapping new nucleotides into a sequence, because it's anabolic, it requires energy, right? The enzyme responsible for that is going to use the energy present in the free-floating nucleotides. Yeah, but we'll only talk about that for like 30 seconds when the slide comes up, unless you want to slow me down when it hits up. Not slow me down, but ask a question. Slow me down is not a negative thing, you know? Like, I'll go real fast if nobody slows me down. I'll just start to ramp. Anyways, I think somebody else had a hand up. Was it you? Yes.
So just to clarify what you said earlier, during the S phase, that's when DNA replication occurs, and then during mitosis or the mitotic phase, replication.
No.
No.
No. Forget I ever said that. I was wrong, okay? I misspoke. I was wrong. I penalized myself by deducting 10 John points from myself, and I apologize, and it's on recording, and I'm admitting I'm wrong and making an earnest apology to you that I will always accept accountability when I make mistakes like that, and if it harmed any of your understanding, I apologize. Please do your best to forget that I ever said that. But a duplicated chromosome is still this chromosome. All right, let's not worry about duplication as a different process, though. That was me making a mistake and being wrong. Yes?
You replaced the word replication with duplication in your notes when quoting the slide. You should revert it back.
I would revert it back, yes. Yep, yes, you should do that. The question was, should you revert that back in your notes if you followed this slide, which is a bad slide, then yes. The slide's not bad. I made a mistake. Because look, this is each duplicated chromosome. I'm like, yeah, but the text lined up wrong, and I got all jumbled in my brain. That's the first mistake I've ever made in my life. It feels weird. Have you ever done one of those? It sucks. No? Yeah. Cool. We're doing pretty good on time. Let's go ahead and take our 5-minute break right now, and then we're going to jump right into the key stages of the cell cycle. All right? So I'm pausing the recording, and I'm going to start a 5-minute timer. We're recording again. Okay, so one of you came up and pointed something out to me. I want to clarify. Oh, I'm going to put my clicker. Oh, thanks. It was obscured by the beaker. Okay. This karyotype, these karyotypes can be confusing because this looks, especially right here, like a duplicated chromosome, right? With 2 sister chromatids. It's not, all right? Each one of these is a distinct chromosome. So there's chromosome 1, and that's chromosome 1. Does that make sense? Let me zoom in on it. So these are not 2 sister chromatids attached at a centromere. They are not. They are 2 distinct chromosomes, all right? This is chromosome 2, and this is chromosome 2. All right? This is what we call a homologous pair of chromosomes. We'll talk about homologous as we move forward. Question, please.
Are those like darker lines somewhat in the middle of the centromere?
You got it, you got it, you got it. You can have 2 points. Yep, that's the centromere. And look, it doesn't pick up stain the same way. Huh, that's interesting. It's because it's this special structure. It's got a bunch of proteins embedded in it. And you want to get really wild with it. There are genes in the centromere that like don't get expressed, usually. And there's also all sorts of interesting stuff with how natural selection operates on genes closer to the centromeres. And there's this was 2 years ago, but there are new aspects of plant breeding and genetic engineering that are just working on getting the centromere to loosen up a little, you know? Because if you can get the centromere to loosen up, then these genes can be expressed or selected for, or transmitted through inheritance differently. It's really cool. The genes around the centromere don't evolve very much because they're all stuck in proteins. It's fascinating stuff. Question?
I was just gonna ask, why are they pairs? Is it the same size?
Because they should be, check this out, right? The maternal side, the paternal side, right? Those 2 individuals, they should have almost the same sequence of DNA. should. They're going to be incredibly similar. You know the shirt people used to wear, like, you're 25% the same genetics as a banana. You know, you're 70% the same genetics as a mouse. You're all this stuff, right? So another human, you're gonna be really similar. So they group them together like this because they're gonna be they're really similar to one another, right? And they're going to have the same genes at the same loci. So that means that, for example, gene A should be here on this chromosome and should be here on this chromosome, all right? Which is going to be key when we start to talk about meiosis and inheritance, all right? And just genetics in general. Did that answer your question? Awesome. Yes.
Just for fun, since you said the Clusinum centromeres kind of thing, is that like a direct correlation that like the common statistics, like, oh, 70% of your DNA is the same as like a banana or whatever?
Yeah.
Is that like the DNA like packed closer to that centromere is more common and that it's more evolved out towards its edges?
I love the way you're thinking, but no. Not necessarily. No, that's not a direct correlation. But that's very, very interesting. It's more that during the generation of gametes, this DNA isn't going to be handled the same as the DNA at the ends. And it's called crossover. We'll talk about it. I'm getting ahead of myself. I'm so sorry. Yeah. But we will talk about it, and I will bring up, it's like that thing I said about the centromeres. And you'll all be like, John, we've talked about 30 different topics since then, and that's okay. Okay, ready? Cell cycle. Here we go. You have to have a mnemonic device for the cell cycle. Everybody has one, right? I don't know what yours is, but I imagine a child at a daycare center, and this child has just during nap time urinated and is very proud of this fact and walks up to their teacher and says, "PMAT, see?" Because they've got a PMAT now and they're showing it off. You understand? All right, very good. Prophase, metaphase, anaphase, telophase, cytokinesis. PMAT, see? There's this intermediate stage called pro-metaphase, which we'll also talk about. But oftentimes, this will just be taught as prophase, metaphase, anaphase, and telophase. Telo, like telo vision from afar. Anyways, great. We're going to talk about these stages. It's going to be great. You're going to love it. Interphase. We ready? We ready to be done with interphase? S is when the DNA gets replicated. And then we're going to enter into M phase. Cool. Okay. So, at G2 of interphase, right before the cell enters into the actual division step, here's what you've got. I told you before, centrosomes are very important. They're incredibly important. They're like microtubule organization centers, and we're not going to talk about them like at all. But you can tell they're important because in this simplified diagram of the cell, there they are, all right? You do need to understand how the mitotic spindle is assembled and what these microtubules are doing. I'll explain it to you. All right, G2 of interphase. Just double back and make sure we know where we're at. The DNA chromosomes have all been replicated, all right? And all the growth is done. The cell is now ready to go into the M phase. The chromosomes are not condensed, but they are duplicated. All right? There is a nucleus with a fully intact nuclear envelope holding all of the DNA inside. Then we enter prophase, okay? The key thing that happens in prophase is the chromosomes condense. So in prophase, chromosomes condense. At higher level genetics courses or cell biology courses, you'll talk a lot about what's going on here with the mitotic spindle because it's genuinely fascinating. It's not genuinely fascinating. It's very dry. But once you're at that level of science course, you'll get excited about all sorts of things that you thought were boring and dry before. There's a lot of interesting stuff happening here with synthesis of microtubules. But there's all these microtubules getting built. Here we go. Key thing in prophase, the chromosomes condense. Prometaphase, the nuclear envelope dissolves. Okay? So you need the nuclear envelope to dissolve in order to attach microtubules to the centromeres. So you can actually move the chromosomes around. How are we so far? So the nuclear envelope must dissolve so that the chromosomes can get separated. The duplicated chromosomes can get separated so that the daughter cells get all the right chromosomes. You had no access here in prometaphase to the chromosomes. All right, prometaphase nuclear envelope dissolves. Okay? If you are taught this with prophase and prometaphase being grouped together, then in prophase, the nuclear envelope dissolves. All right? And I'm sorry to, have to give you both definitions or both classifications or both schemes for understanding this at once, but it's talked about both ways. Prometaphase doesn't always come up. So,
What did you say?
please, yeah.
You need the nuclear envelope to dissolve to attach the microtubules to the centromere?
Nailed it. Points. Yeah, you get points. You can have. I think you deserve 2 for putting it so eloquently. Yep. The nuclear envelope dissolves so the microtubules can attach to the centromeres. Okay. Cool. Now, when the microtubule attaches to a centromere, now it gets a new name. And now it's called a kinetochore, like kinesiology, movement, right? The kinetochore forms, and that's going to allow the cell to move around the chromosomes. I've got another diagram of it, if it's helpful for you. I'm just introducing it right here first. So the kinetochores form also in prometaphase. Those microtubules grow out, they attach to the duplicated chromosomes, they attach to the centromeres, forming the kinetochores. The microtubules that attach to the centromeres to form the kinetochores are called kinetochore microtubules. All right? Because there are other microtubules that are also growing out. So there are non-kinetochore microtubules in this process, and there are kinetochore microtubules. What are the non-kinetochore microtubules doing? Check it out in your textbook or don't worry about it. All right? We got way too much complicated stuff happening to overwhelm you with that. Cool? All right. Awesome. We're good so far? Now that the kinetic, yes, please.
I appreciate this all quite simplified. But if we're dissolving membranes and boundaries, are there any significant? Does the cell as a whole shift its pH or its saltiness to accommodate that?
Oh, I mean, as soon as the nucleus and the cytoplasm sort of blend together, they should be similar in their pH. It is not through differences in pH that DNA gets denatured or broken down if it falls into the cytoplasm. It is through the activity of special enzymes and nucleases. All right? Now, those nucleases, those are going to need to be inactivated, right? But they also don't operate the same on condensed chromosomes as they would on uncondensed chromatin, right? So the cell does need to make changes because normally, if there's DNA in the cytoplasm, you want to destroy it because it's a virus trying to infect you. Yeah, make sense? Sick. But the solution should be similar in pH. I'm sure that there are differences between the nuclear solution and the cytoplasm, but yeah. Awesome. Okay, now that the kinetochore microtubules have attached and the kinetochores have formed, all of the chromosomes, which remember are duplicated, okay, they're all going to align at the center of the cell in preparation for being yanked apart. They align at the center, and there's not a real structure here, but they call this the metaphase plate, all right? And this step is called metaphase. Just think meta like middle phase, even though that's not the correct root for the word meta, but don't worry about it. You can just think M, middle, all right? Metaphase. All of the chromosomes have aligned at the center. Remember, they are duplicated chromosomes made up of two sister chromatids. So at the next step, they're going to get yanked apart, all right? And that's going to generate two chromosomes. Two chromosomes from one duplicated chromosome. How are we so far? I know I'm really going slow on this one, but that's intentional. Like I said, 50 slides we have to get through in 4 days, so we can take our time. Can I clarify anything else on this slide so far? Metaphase is the friendly one. When you see it on an exam and you're like, and it's like, identify what stage of the cell cycle this cell is in, and it's this, you're like, yes. Metaphase, easy, no stress. I don't have that question on your exam, I don't think, but you'll have it on a genetics exam if you continue in the course, in the track. Anyways, next we've got anaphase. All right. This one's friendly because the chromosomes look like little A's, kind of. All right? That's at least how I always remembered it. Here's what's going on. All right? The sister chromatids attached at the centromere, okay? The duplicated chromosome gets all aligned at the middle with kinetochore microtubules attached. And then when they get pulled, they're being pulled through a fluid. So imagine I have a rope tied to my belt, right? And I'm like hanging out, and there's another one of me, and we're just like face to face, like just talking, like la 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 la, and then we kiss. No, we don't. All right. And then this rope gets pulled. So now we're hanging out, and then the rope gets pulled. I go, woo, like that. All right? That's the shape my body would make, and that's the sound I'd make. Okay? And that is the sound that these chromosomes make. All right? So anaphase. The sister chromatids get separated, and now the unduplicated chromosomes start to get pulled to the poles of the cell. They start to get pulled to opposite poles. Confusing, but that's the language that's always used. All right? Now, of course, you're sitting out there, and you're like, John, microtubules aren't designed for pulling things. You told me way back in week 2 that microtubules are better for compression resistance and not good at pulling. And I'd say, good, great. I'm so happy that you remember that. But this time, they are used for pulling. And it has to do with how they are synthesized and desynthesized at the asters. And as I said, we are not going to worry about it. We're not going to trip about it. It's fine. It's fine. All right? Cool. There's anaphase. Anaphase is another nice one when you get it on an exam because you're like, oh, cool, they all look like little A's. Anaphase. Awesome. Good? The last one is telophase and cytokinesis. It drives me nuts because they used to teach, back when I was a student, I got taught this process with no prometaphase, but with telophase and cytokinesis split into 2 steps. But now the textbooks like to break out prometaphase and group together these. Here's what telophase and cytokinesis are. In telophase, all right, remember telo like from afar, like the chromosomes are now afar. In telophase, the nucleus begins to reform, the nuclear envelope reforms. This is nucleolus forming. If you remember, that's about that's about ribosome synthesis. Don't worry about it. I say don't worry about it a lot. All right. Nuclear envelope reforms. Okay. Now the chromosomes are fully not going to mix up. Cytokinesis. Okay. In cytokinesis, you actually get those cells to split. This is accomplished with an actin filament, an actin microfilament ring that sort of spans around this. I'm not going to test you on that, of course. Okay. But it's like a string, wrap it around a balloon, pull it tight, the balloon splits into two. Fair? Okay. And thankfully here, it's actually a filament, which is, for pulling things. Okay. It's frustrating that microtubules are used for pulling things in the cell cycle. But like I said, it's because of how tubulin gets polymerized. And that's the name for the monomers, the proteins that make up microtubules. They named them tubulins, which is weird. It's not weird. It's just obvious. Okay, that's PMAT, see? Here is this is a cell in metaphase. To get to metaphase, all of your kinetochores had to form because the kinetochores are what let us move the chromosomes around, right? Very good. Remember, kinetochore, that's the structure of the centromere with the kinetochore microtubules. All right? Here's the metaphase plate. There are non-kinetochore microtubules. What am I missing? I'm missing another piece of terminology that overlaps, but one which you need to be familiar with because in literature and in publications, they usually don't refer to kinetochore microtubules. They'll refer to the mitotic spindle, which is very frustrating. The mitotic spindle is the structure of all these different microtubules attached. The mitotic spindle is all the microtubules that are attaching to a kinetochore. All right? So we call them kinetochore microtubules, but then there are multiple ones, and all of them grouped together can be called a mitotic spindle. It's really frustrating. Yes?
Mitotic spindle there, isn't that coming from the non-kinetochore microtubules?
This is coming from the kinetochore microtubules. This is made up of kinetochore microtubules.
I'm still trying to distinguish the non-kinetochore.
Oh yeah, no, it's super freaking confusing. The non-kinetochore microtubules are those that do not connect to the centromere. You can see them right here. See how they're involved in other things are moving around. All right, the microtubules are good as little highways for moving stuff. You have to sort of sequester organelles and things like that towards the different poles. So that's happening there. There's all sorts of important stuff. But we're just going to worry about, I'll tell you right now, we are focusing on the cell cycle from the perspective of getting the DNA where it needs to go. All right? Awesome. So I always hate to have to give you overlapping terminology, but please be aware of the mitotic spindle. Mitotic spindle microtubules, kinetochore microtubules, for all intents and purposes, same thing, all right? But if you're reading literature that's talking about cell division, and trust me, eventually you'll have to, if you're in bio at all, they will probably say mitotic spindle. They go, the absence of this gene interfered with the mitotic spindle polymerization or formation. Kinetochores could not form because the mitotic spindle did not extend at the rate that was required, as determined via fluorescence and blah, 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 blah. It'll be a really exciting paper, I'm sure. And hey, man, if you want to be a biologist working in a microscope, staying on the cutting edge, always having grants, always having work to do, study the cell cycle. This is money. This specialization is just bio money in the bank. Like, you can get better and better and better and better at it. It requires all these different microscopy techniques. You can get more expensive equipment with grants. And some of that grant money goes right to you. A lot of it goes to the university, but some of it goes to you. Anyway, studying the cell cycles really just, that's what I was gonna do when I was here. And I was trying to go for the tenure track. I had a whole lab research plan set up. And then I just met too many people who were tenured faculty. And you meet enough of them and you just realize, oh boy, I really, what am I saying? It's on recording. They're great people, but they're tired all the time. Research isn't for everybody. But if you love the idea of microscopic research, cell bio research, studying the cell cycle's a great option. This is just genuine advice. All right? There's tons to be done in that field, and we understand very little of it. And the more we understand, the research in the cell cycle is how we're developing the most effective cancer treatments. All right? It's all through cell cycle research. And really, it's all about these dang microtubules. That guy who bought the LA Times, that scientist, he did it because he figured out how Taxol worked. And Taxol interferes with mitotic spindle generation and attachment. And then he made millions of dollars. And Taxol works to inhibit cancer without, as drastic a chemotherapy. I don't know. You know what? Screw it. It's interesting. I'm not going to pretend it's not. Study this stuff. And then give me the credit for encouraging you to study this. And then I get to feel like I did something great in the world when you resolve some sort of, when you cure some sort of cancer. Thank you in advance. All right, chromosomes, microtubules, microfilaments. Here it all is in a picture. Everything's stained for you so you can see what it actually looks like. All right, there's microtubules involved, there's microfilaments involved, all the stuff we've talked about. Questions. Here's all the phases in images. Cool. If you want to get a leg up on doing this sort of research, if you're like, you know what, I'm inspired, then you can talk to Dr. Amy Sprouse, who is incredible on campus and has access to the equipment you need to collect images like this. Question?
On one of the prophase slides, there was like an arrow pointing to the early mitotic spindle, and I can see.
Oh wait, should I not be going away? Sorry.
Well, I can see that it's the same thing. It's like the neon green.
Yeah.
So then, how does it move from right there to be on two different?
To be on both sides. It's a whole field of study.
Okay.
Yep.
Okay.
Microtube.
I don't want to concern myself with that.
I
right
would
now.
say don't.
Okay.
The aster, here we go, right here. The centrosome,
Yeah.
the centrioles, the asters, all of this is its own field of study and research.
Okay.
And it is incredibly important. And yeah, it's got great job. You got a point for that. Because look at it right there. It's blasting out. It's getting ready to split off. Yeah, it's pretty cool. And then the key thing is each daughter cell has to end up with an aster so that it can get a centriole. That's also incredibly important. If that doesn't happen, you make a daughter cell without a microtubule organizing center, the cell can't divide anymore. If the cell can't divide, it can't be cancerous. Pretty good, huh? Yes, please.
So, is an aster what exactly an aster is?
It is a centriole when it's got microtubules growing out of it. It's called an aster with the same root as asteroid, meaning star. It looks like a star, as you can see in these images right here, where it's a total aster, no doubt. But yeah, like I said, for this course, don't focus too much brain power on that stuff. But boy, oh boy, is it interesting. Okay, moving on. Cytokinesis, okay? Remember, telophase, the new nuclei start to are reforming, okay, to separate all the different chromosomes. In cytokinesis, the cytoplasm moves. Cytokinesis is just splitting of the cell. And it is different in animal cells versus plant cells because of the presence of a cell wall in plant cells. So in animal cells, it's just like a balloon. I was gonna just blow this glove up, but I don't know where it's been. I'm not gonna put my mouth on it. All right? You can imagine a balloon. You've seen a balloon before. In an animal cell, it's like a balloon. Here I go, I've got it right here. I twist it, all right? And it makes a cleavage furrow there. And then it just pops off, all right? Split like that. Because there's no cell wall. In the plants, there's a cell wall. The cell wall is made of cellulose. Very, very good. All right. A cell wall needs to form between the two cells. So basically what happens is the whole thing is the whole process of prophase, metaphase, anaphase, telophase occurs, bless you, inside of the box of the cell wall. The new nuclei start to reform, and then cellulose just sort of gets deposited along this plane, all right? Cellulose gets deposited, it forms this cell plate, which then attaches to the cell wall, making a new cell wall, all right? So it's an additional complication. And then you're saying to me, but John, doesn't that mean that during mitosis, a plant cell just goes like this? It forms new nuclei, and then it forms new nuclei, and then it forms new nuclei, all right? Eventually just getting smaller and smaller and smaller. And I would say to you, well, no, all right? But good thinking, that's what would happen, except, folks, that there are some enzymes that come into play here that sort of loosen up, all right? They loosen up the cell wall and allow it to expand via turgor pressure, okay? So the cells in a plant don't have to just get smaller and smaller and smaller and smaller, all right? But the key difference between cytokinesis in animal and in plant cells is you gotta make a cell wall. Questions? Very good. Question, please.
It's really interesting about a lot of like the chemistry level physics.
Yeah.
Specific molecule structure reason that we see the geometry that we see in cell walls, like that they have to be like split in square geometric ways.
It more evenly divides the cytoplasm so that each daughter cell is more similar to one another. And remember that the structure of the plant is determined to a large extent. Well, I'm going to say the structure of the plant is determined by the cell walls, right? They give it the structure. So if you had them being divided like in weird directions and breaking off and would just be different sizes, it'd be like trying to build a brick house with a bunch of bricks that are different sizes, which I'm aware could be done, but a plant's, not gonna do that. That makes sense? Sick. And molecularly, yes, there are ways that these things get targeted to these specific locations that I did have to learn and I have forgotten. So my apologies. Question, please.
What is SEM and what is TEM?
SEM is scanning electron microscopy. It just shows us surface images of structures. It scans the surface. TEM is transmission electron microscopy, where the electrons actually go like that through samples and let us see internal structures. It's analogous to using a dissecting microscope versus using a compound microscope. except that they're both capable of similar levels of magnification. Nice, yeah. And we've got both on campus. There's classes where you can learn how to use them. And if any of you remember our last visit to the core, Liz, who is the core manager, she does a lot of SEM stuff. And she could totally, if you just go up and talk to her, she could help you work out a time to go use the SEM probably, which is cool because it's an expensive piece of equipment. It's neat. You have to gold plate samples too. It's really cool. We have a bunch of gold plated spiders upstairs. People seem to think that's interesting. I do. Yes.
Do you have to have a specific project to do that, or can you just go up there and be like, hey, can I look at something cool?
Totally just go up there and be like, hey, I want to look at something cool. That's part of our job here, and I think that we forget it sometimes as professors, as lecturers, as college level instructors. It's like, it's still okay if you just want to see something cool. Yeah, like if you just want to go up and look at the gold-plated spiders, you can do that. You don't have to actually ever even look at an SEM. Or you can go up there and be like, hey, I want to get into microscopy a little more. And Liz would help you out and maybe put you in touch with some people who do more of that stuff. We got really cool stuff in the core. Highly encouraged. Yes, very good. Okay, let's keep on moving. We're going to continue moving forward. We're making really killer progress here. All right. I'm going to drink a little more coffee. We're going to keep looking at the cell cycle. Okay, we're zoomed back out again. So we're looking at interphase and M phase. So we're including G1S, G2, and there's M. This molecular control system, we are going to cover it at a very basic, very simplified level, okay? With the discussion of checkpoints. It's an interesting diagram that a lot of textbooks use. It's like you imagine that this bar is spinning, and if it hits one of these gates while they're up, it'll stop, and the cell won't proceed into the next stage. See what this is supposed to look like? Like you drop these down, you raise them up. You ever put a light on one of those mechanical timers that spins and you raise or lower little tabs, those things? They make sprinkler systems that work that way too. That's what this is attempting to mimic. But those systems now are mainly digital. So it's one of those things about like using a typewriter as an analogy for you. I'm like, you gotta put new ribbon in the typewriter. I'd be like, yeah, sure. Yeah. But, that's what this is mimicking. Here's the key takeaway. There are 3 main checkpoints that the cell has to sort of pass through in order to complete the cell cycle. And these checkpoints exist to prevent the cell from replicating from duplicating, from producing 2 daughter cells before it's ready. All right? You've got a G1 checkpoint, you've got a G2 checkpoint, and you have a checkpoint within M phase. We're going to talk about one example of how this can work with with cyclin-dependent kinases and cyclins. All right, we'll talk about that. But there are a lot of different things going on here, of course. So, the presence of different molecules, all right, allows the cell to move past these different checkpoints and complete the cell cycle. The G1 checkpoint. It is located in G1 of interphase. The cell checks to see if it has enough resources and if it is of an adequate size. The cell also checks to see if there is DNA damage. All right, how does it do it? There's a bunch of enzymes. All right. It's not entirely accurate, but it's close enough. you know? Yeah, if you can't tell, I don't like. I always have a problem with these very surface level like explanations, but the cell checks for adequate size and resources. Because if there aren't enough, if it's not big enough and it doesn't have enough resources, you definitely don't want to enter S phase because you're not going to be able to carry out all your transcription and translation as the chromosomes are busy duplicating. Make sense? If there's genomic DNA damage, that's bad. You do not want to be replicating damaged chromosomes. You don't want to be replicating cells that have damaged DNA. So cells have all sorts of internal mechanisms for checking that, and they'll try to repair even. The cell can even repair some damaged DNA. But if it can't repair, it will trigger apoptosis, which is programmed cell death. Okay? Question?
What about mutation? Is that like successfully not say it's always good, but
Yeah.
if it's the cell stops dividing or like when the mutation occur, if cell death occurs before it gets the chance to pass on those genes.
So oftentimes mutations just get past these. These systems are not foolproof. They don't always detect genomic DNA damage.
Mutation doesn't mean damage in sense.
It just means a change in nucleotide sequence. It specifically changes in, it was AAAT and now it's ACAT, you know? And also some of these screens for genomic DNA damage are very basic of like, is the DNA the right width all the way through? Because if there's a mismatch somewhere, they'll make a bubble in the DNA or it'll make the DNA pinched in. And some of these checkpoint enzymes, all right, are just scanning the DNA and measuring its width. So that can totally miss if a purine gets swapped with a purine. You know what I'm saying? Sick. Okay, very good stuff. Moving forward, if it passes G1, DNA gets replicated, S phase happens, right? You enter G2, which is right before entry into the mitotic phase or into M phase, so right before prophase begins. So once again, making sure the cell has grown and is of an adequate size. It's also checking, though this one is also checking that all the chromosomes are replicated, okay? Because if the chromosomes aren't all replicated, you do not want to enter into mitosis because you would end up with one daughter cell missing a chromosome. You see how that sort of makes sense? The chromosomes need to all be replicated because if there's an unreplicated chromosome, then one of your cells, the kinetochore microtubules attach, and usually it goes like this. But if it's an unreplicated chromosome, they still attach, and it just goes to one pole. Then the other cell is missing a chromosome. Cool? Awesome. Oh, hey. So there's an entire topic that I cut in the summer because we did have limited time. And it means that there's one thing, apoptosis, which we don't really talk about. Apoptosis is a very controlled, programmed cell death where the cell is destroyed in a way that keeps adjacent cells safe instead of just exploding the cell or something. There's a whole section on cell signaling, all right, that we don't really talk about because it's just a lot of like, this receptor works like this. What does it do? A lot of different things, okay? But it means that normally you would have been introduced to the word apoptosis. So I just want to make sure that you've heard it before. Okay. Cool. Cool. Okay. Very good. Then there's an M phase checkpoint. All right. The M checkpoint. This one checks that the mitotic spindle has attached to all the chromosomes. So all the kinetochores have formed successfully. Because imagine you've got a duplicated chromosome And if the kinetochore microtubules only attach to, say, one side, like on my left, your right, all right, then the entire duplicated chromosome gets yanked over to this cell. And now that cell has an extra chromosome, and the cell over there is missing that chromosome. All right? So errors in these checkpoints, errors in microtubule attachment, that's primarily where we get abnormal chromosome numbers, like multiple copies of a single chromosome or missing copies of other chromosomes. All right? Questions? Yes?
So, I don't know if I'm completely understanding the diagrams. It looks like
Yeah.
the M checkpoint is after prometaphase, and then in the second one, it's pointing at metaphase, and it looks like, I can't tell if it's before or during the checkpoint.
Oh yeah, it's like during.
So
Should be right in the middle.
the checkpoint is basically at metaphase.
The checkpoint is basically making sure prometaphase completed successfully.
Okay.
And then once prometaphase is fully completed, metaphase can sort of happen. This is another one of those problems of grouping, of separating these stages out more and more, which I'm kind of against, because when you're trying to deal with foundational concepts, you could take this process and separate it into 30 steps if you wanted to. You could have it, you know what I mean? Like, and they do if you're studying this process specifically. But yeah, the key thing here is that in order to pass the M checkpoint, kinetochores need to be attached, need to be fully formed. Mitotic spindles need to be fully attached. Okay. One way that we, oh yes, question, please.
The first checkpoint, the G1 checkpoint, you said it goes into G0.
Oh yeah, right here. Yeah, G0 is just like, you know how there's G1 and G2? G0 is like, stop doing anything, cell.
Do
All
the
right.
other checkpoints, if it doesn't pass, go somewhere?
The cell can go through apoptosis or this, but the cell will usually just try to correct that. Yeah. This is the key one where it gets shunted off to this G0 phase, where it can still persist. It'll still do stuff. Because think about it this way, right? It's got all the chromosomes. They're not all replicated, all right? So it can still do its job. You know what I mean? And it can sometimes enter out of G0, you know? So yeah, you get pushed into G0 for a while, and maybe you're able to be repaired. It's like pulling over. You're a NASCAR car going in a big circle. And you get pulled over into G0 where you're waiting, see if it can get fixed. But you can still do the important things that a NASCAR person does, which is drink milk, I think.
If it doesn't pass the other checkpoints, it goes into apoptosis.
I want to say don't worry about it, but in this case, that's more of a cop-out for me, of like, I don't have enough information, I don't know enough to give you what I would consider a satisfying answer to that question. Yeah. That's twice today. Had to admit I didn't know enough. All right, very cool. I'm gonna have to deduct you points though. Yeah, because you made me look like a fool in front of the entire class. Okay, I told you we'd end early, and I totally forgot until just now. I think technically our class does run until 12:05 though, so we're gonna end on this slide right here, and we will come back because we have to talk about this specific molecular regulator of the cell cycle, which involves an enzyme class that we haven't been introduced to yet, called the kinases. It would normally have been introduced in the cell communication slides, but it's one of the 3 things that I think are actually essential from there, so I'll just emphasize it for you tomorrow on Tuesday's lecture. Okay, so that wraps up today. We are on slide 21 of 50. So we're making killer progress. We'll go all the way through this stuff, which includes more DNA, which includes some DNA replication. It's going to be a lot of fun. And yeah, we can talk about it as much as you want. Anyway, that's the end of today's lecture. I have your midterms at the front. You can come and take a look at them. I'm going to stop this recording now.